Good morning, good afternoon, or good evening, wherever you are to everyone. This is Banu Cannon. I am in CDER's Office of Compliance and Biologics Quality Bioresearch Monitoring Branch. Today, I'm going to briefly talk about the perspectives from CDER's Office of Compliance and Biologics Quality and our experiences with data submissions to CDER. So briefly, the agenda is to talk about the data standard requirements. The other speakers have already touched upon this, so I will be very brief on that, and our CDER Bioresearch Monitoring Inspection process with the submitted data, our review of the submitted data, and experience and challenges with the submitted data. And finally, a few suggestions for sponsors, and last but not the least, FDA resources. So, study data requirements. Why should submitted data be standardized? We all know standardization helps us. For instance, if you have children, if they have taken tests, we can compare the results. So standardization will lead to comparison of results. So here, this will help streamline FDA review process, and it enables consistent use of the analysis tools. During the review process, we use various tools, so this enables consistent use of such tools. And this is briefly some of the references that I know the other speakers have provided that you can refer to. And as the previous speaker mentioned, the CDER study data submissions link may be revised, or you may want to talk to the regulatory project manager as suggested by the previous speaker. So, what do we do? This is our CDER Bioresearch Monitoring Program and our process. So what does our branch do? We conduct pre-license and pre-approval data verification inspections. This is one of our program elements. This is where actually we do inspections of the data submitted to the BLA or PMA or supplemental BLA. Then we also investigate complaints that come to us from the sponsors, or sometimes it could be consumers or clinical investigators. We also answer questions about good clinical practice, GCP. And we help evaluate concerns about data integrity, namely the entities that we inspect. They are either clinical investigators, sponsor monitor, or contract research organization, or CROs, or institutional review boards and good clinical laboratory. And the goal of all this is to namely the public health mission, which is to protect safety, welfare, and rights of the human subjects who participate in the clinical study. So this was shown by the previous speaker. This is the 21st century review. And this is for the 10-month review clock. And for the 6-month review clock, everything will be even shorter. So this shows briefly our review process. So shifting gears, this is our BIMO review process. Where do we stand when the biologic licensing application comes to CDER, our door? And as I said, the 6-month clock is adjusted accordingly. And so this is I'm going to concentrate on the lower half of the cartoon, or the visualization of this particular slide. So first, we receive, let's say we have meeting. We participate in the pre-BLA meeting along with the other review committee members. So in CDER, we in Bioresearch Monitoring Branch participate during the entire review process, from the start pre-BLA meeting all the way up to just before the action due date. So during the pre-BLA meeting, we may provide comments. And next, the data comes in, day 0. And then within roughly the first month, so the first committee meeting, which is around 30 days, we review the data. So this is very critical. So for our review process, namely for our review and inspection process, we have to review data within the first 30 days. So you can imagine if the data comes in that is not standardized, it will be very difficult for us to go through all the data and go to the next step. So after we review the data, and then we have filing meeting, and roughly within the first 60 days, we issue the inspection assignments. So for this, we go through standard procedures and go through our process, and we issue the inspection assignment. So within the first 2 months of the 10-month review clock, you can imagine we have to go through all this data, select sites for the inspection, and especially if it is a bigger vaccine study, as was alluded to by the previous speakers, this is a huge task for us. So we go through all the data, select sites for the inspection, and then we send it to the particular Office of Regulatory Affairs field investigators, and they conduct the inspection. So after that, depending upon the workload, we will receive our inspection reports after the inspections have been completed, which may be roughly after 90 or 120 days after we issue the inspection assignment. So after that, we review the inspection reports, and then we draft a summary, our recommendation to the committee based upon the inspection results. And we also write letters to the inspected parties. So this gives an overview of our process. So next, I'm going to concentrate on the site selection. So for all this to happen, we have to select the sites for inspection. So what do? The first process is, as I said in the previous, from the previous slide, we receive data from for the BLA or SBIA or whatever. We receive the data tables. And I also want to reiterate the point that we work across the product offices in CDER. So we work data that comes into CDER, that comes to Office of Blood, or it could be Office of Vaccines, or also the new reorganized office, Office of Tissue and Advanced Therapies, which also include products under cell and gene therapy. So we work across the various product offices, and we review the data. So we receive the data tables, we review them, and then when we do the review, we also review the companion protocol, and along with that, the study report and all the data that comes in through the gateway. So there is a lot of information that we have to review before we draft our assignment. And the examples of data that we normally verify at the clinical site, adverse events, protocol deviations, and definitely the efficacy and safety endpoints, blinding, these are some of them. And pretty much we can understand from this that probably all the data that goes into the label that the sponsor has used, we will try to verify at the clinical site. So continuing with our site selection process, what else do? We consider other factors in site selection, which include the data you already submitted in the BLA, which is subject disposition, and also exclusions and discontinuations, namely screen failure data or any discontinuation that occurred during the study. protocol deviations, and other data that I had mentioned earlier. We use these factors to select sites. And the second half of this comes from our internal database, namely, this is the inspection history of investigators and any good clinical practice GCP problems reported by the sponsor, and the number of sub-investigators. And last but not the least, the pending workload in the FDA geographic region who are actually conducting the inspection. So I want to briefly state that in CDER, we do not have any site selection tool to help us with this. The second half of this, the inspection history, GCP problem, all this come from our internal database, and this is a manual process. We go through that. So what do we compare? We compare the source data at the site with the data that was submitted in the BLA. And this was, the earlier speaker had talked about the data, namely we have the data, like it could be all the adverse event, the demographic data, the dispersion data, whatever that was submitted in the BLA, we will compare with the source data. So that's why we want raw data or the study data tabulation model, or rather SDTM data. So that's the data we review, not the analyzed or the derived data, which is the ADAM analysis data model, ADAM data, because we are comparing the raw data at the clinical site. And this raw data or the source data at the site could be doctor notes, progress notes, nurses notes, or it could be medical charts from the hospital. So we compare any of the source data with the data that was submitted in the BLA. And it could also be electronic source data, because we know more and more these days a lot of data comes in as e-source, so we do verify them. So it could be electronic case report form or any source data that is available at the site, we will verify. So what are the challenges? In our experience, we have seen some of the challenges which are explained in the following few slides, but this is just a synopsis of all of them. One of them is site ID variable. It was not always included or available in all data sets except the demography, the DM data set. And for us, as I had mentioned in my earlier slide, we have to review all the data within the first 30 days, which is a lot of data for us to review. So if the site ID, because we are selecting sites to inspect, if the site ID variable was not included in some of the other data sets, it's not faster for us to select the sites. Let me put it that way. So we want to facilitate our site selection. So we would prefer the site ID to be available in other data sets as well. And we have seen examples wherein the data set submitted did not include standardized variables, or the deviations were submitted as PDF listings. So I'm going to give some examples for this. And the previous speaker alluded to this last bullet, namely rows of data were missing. This was adverse event data. We do not know if this was due to inadequate data validation or what the reason was. And this creates issues for us. And obviously, there are more surprises if there were no pre-BLA meetings. So this is one of the examples wherein we had a sponsor submitted, instead of the deviation DV data set with the standard variable, Here, the sponsor had submitted the deviation as a different data set. And please note here, instead of the subject ID, it is represented as PID. And there is an LM code which probably is the deviation code. And they did not provide what it was. Instead, the sponsor submitted a companion Word document to tell us what those 111 or 112, what those stand for. So all this will make the review process a little longer for us to select the sites. So there were non-standard variables that were used. And this is the next example wherein the protocol deviations were submitted as PDF listings. Imagine in this day and age, if we have to go through 3,000 to 4,000 pages of PDF listings and pick one site or pick 4 or 5 sites for an inspection, this is an enormous task for us to do. So this was PDF listing. And also, if you look at the last row in this particular PDF listing, the sponsor acknowledged that there were issues in the integration of the different computer systems used during the data collection. So we do not know how much we can trust if they already told us that there were problems and when we start to select sites for inspection. And in this example, again, instead of use subject ID variable, the sponsor had used patient. And if you look at the recent one, you can see that this was incomplete information. They did not tell us why this particular subject was withdrawn. And without complete information, it is difficult for us to understand what happened at that particular site with this particular subject. And this was just one example. This was prevalent throughout the submission for us. And the next example we have is where the protocol deviations were submitted as comments. So instead of the traditional deviations, these were submitted as comments data set. And as you can see, it was a different domain. So there was no deviation data set to tell us about the protocol deviations. And so this was very difficult for us to select the sites again. Again, it's just all this have to be done within the first month or so. And in another study, we noted that there were lots of variables. However, the variables did not have any definition, so we had no idea what those variables stood for. And this is an example wherein the definition file, I know earlier Ron had mentioned this and other speakers had alluded to, we want the definition file to be user-friendly. We want to know what is in the definition file. And this is one of the good examples. Namely, here the DV data set has all the information, the definitions, and it's easier for us to select the sites in this particular study. And the definition file, this was one of the good examples wherein we could easily go to the particular definition file. We know which case report form page we have to refer to. So this was one of an excellent example of the definition file we received. So what is the suggestion for good quality data submission? We request the sponsor to develop protocol-specific case report forms. Other speakers alluded to this, and also annotated case report forms. They are very useful, and if you can include them, that's what I think is required. So study-specific data collection. If the protocol specifies you need to collect on such and such a data and such a date, the data should be collected according to the protocol. And this was referred to earlier too, advanced discussions with CDER, especially if you are using e-source data collection methods or if you are extracting data from electronic data capture systems, we request that you have advanced discussions with the particular product office that you are interacting with. This will definitely expedite our inspection process and site selection. And advanced discussion about the study data standardization plan. This will again help us tremendously. Last but not the least, the good quality data that comes through could potentially avoid delay in our inspection site selection process and help you all. So this is a slide that you may find it very useful. Thank you for your attention. Now I'm turning it back to Brenda Stodden.
Many thanks to our speakers and also to the audience for your attention.